Hello and welcome to our presentation about the use of DNA fingerprinting in forensics. DNA fingerprinting was the first molecular genetic forensic technique developed. The aim of this technique was to identify with as much certainty as possible the origin of a biological sample. There were many discoveries in science that led to what we know today as DNA fingerprinting. The first major step towards the discovery of this technique happened over a century ago when Carl Landsteiner discovered the human ABO blood group system. This discovery led to the ability to rule out suspects conclusively by looking at their blood grouping. Between the early 1900s and the 1980s, other influential developments were made, including the first antibody test for blood group polymorphism and the Locard-Ley principle that every contact leaves a trace. In the year 1984, at Leicester University, Alec Jeffreys and his graduate students were researching the evolution of globin genes in sea-dwelling mammals. The results of one of their experiments yielded an unexpected but decade-defining result. Jeffreys and his team noticed that a particular 33 nucleotide sequence appeared in a variety of repetitive patterns, and that they were present in the genomes of most mammals. It was decided to test to see if the potential markers, mini-satellites, occurred in the human genome. Jeffreys and his team believed that the mini-satellites would pinpoint genes that were in fact responsible for different traits within each individual. Early one September morning, later that year, Jeffreys and a graduate student observed the results of their experiment on a human genome. Much to their surprise, probes had picked up several mini-satellites, but in addition to this, the many satellites appeared to be uniquely distributed among individuals and passed down through families in medallion fashion. Jeffreys and his team submitted a publication to one of the world's leading multidisciplinary science journal, Nature, early the next year. Word quickly spread that this new technique could prove the origin of a human sample. Despite the fact that the technique was still in development, it was used to support high-profile court cases, resulting in conclusions that were favourable to the DNA results. DNA fingerprinting became in high demand due to the favourable outcomes. Jeffreys was inundated by requests for this test to be used in many other circumstances. Advances and developments needed to be made, along with the standardizing of the method to increase the validity of the technique. In Jeffreys' original method of DNA fingerprinting, hybridized probes detected mini-satellites made up of tandemly repeated sequences. Though this method was effective, it did mean that a large amount of DNA was required for accurate fingerprinting. This meant that there were numerous samples that were too small, contaminated, or damaged to use. One of the improvements that was made was the process of amplifying the sequence using the polymerase chain reaction, PCR, which increased the sensitivity of the test, allowing the use of small degraded DNA. Today, the same method applies, but it is now done by automated sequencing technology, which uses 4 simple short tandem repeats, which are in uninterrupted runs of a single repeat type, which offers faster results that are accurate even with mixed samples. So how does it work? The process of DNA fingerprinting works by collecting a sample of DNA, treating it appropriately, and then running the sample through a process called gel electrophoresis to obtain the individual pattern from that DNA. Deoxyribonucleic acid, DNA, is a genetic code that contains the information that living organisms need to grow, reproduce, and function. This information is stored in the nucleus of eukaryote cells and is unique to each human, with the exception of identical twins. The building blocks of DNA are nucleotides, which contain 3 components: a nitrogenous base, a 5-carbon pentose ring, and a phosphate group. The nitrogenous base is one of 4 bases: adenine, guanine, cytosine, and thymine. The nitrogenous bases are divided into 2 types, purine and pyrimidine, depending on the number of carbon chains that it contains, with purine containing a 2-carbon nitrogen ring and pyrimidine containing 1-carbon nitrogen ring. The base will match with a complementary base, adenine with thymine, and cytosine with guanine. The process of DNA fingerprinting requires the collection of a sample of DNA, usually tissue, semen, saliva, or blood. The sample is cleaned using a range of chemicals which leave pure DNA, which is white and viscous. After being stored in water, restrictive enzymes are used to cleave a DNA at particular points. The DNA is cut at specific letter sequences by the enzymes. The sample is then put into a gel electrophoresis machine. The process of gel electrophoresis requires the sample of DNA to be put into wells at one end of the agarose gel. Agarose gel is a polysaccharide polymer that DNA can pass through. When the electrodes connected with the gel are turned on, a current will pass through the gel. A negative charge will occur near the wells at one end and a positive charge at the other. The phosphate groups on the nucleotides are negative due to the negatively charged oxygen in their group, making them move through the gel towards the positive side. The size of the DNA strands affects their rate of movement through the gel, as smaller strands move faster than longer strands. As each person's DNA is different, so will be the pattern that their DNA makes in the agarose gel. It is at this stage that a dye can be added to stain the samples to make the proteins, which are individual to each person, visible. The separated DNA fragments look more like a smear at this stage, so the sample is exposed to a weak acid to create the lines and then transferred onto white nitrocellulose paper. This is southern blotting, which transfers the DNA onto a membrane, and then a radioactive probe is applied, and the membrane is then exposed to X-ray film, which creates the barcode-like pattern generally associated with DNA fingerprinting. These results can be compared with those of other suspects to determine the origin of the sample. And that's it from me about the history and development of DNA fingerprinting and how it works. Now Megan will discuss the advantages and applications of this technique.
Hi, my name is Megan, and I'm here today to talk about the applications and advantages of DNA fingerprinting. The applications of DNA fingerprinting are diverse and extremely useful. It can be utilized for criminal investigations to prosecute a suspect, paternity testing to identify a biological parent, and DNA testing can also be used to confirm or rule out a medical diagnosis. It can also be used for animal pedigree verification to determine, for example, if a dog is a purebred, and in identifying deceased or missing people otherwise not identifiable under usual means. The capability of DNA fingerprinting in homicide investigations can be invaluable when DNA analyzed has linked a suspect to a victim. In one observational retrospective study that investigated the role of DNA in homicide investigations, found that the husband that previously had a solid alibi was found guilty of murdering his wife. Detectives found small traces of blood on the husband's shoes and when tested were linked to his wife, and this ultimately led to a successful prosecution. Additionally, assuming there's sufficient DNA, cold cases can be revisited and previous prosecutions can be revised, and wrongly convicted people are able to walk free. Another capability to utilize DNA testing is to identify the deceased when physical identification is impossible. For example, a disastrous event involving a car pile-up in a highway in Germany where tragically 8 victims died. They were classified as high-grade heat-damaged victims, and their remains were beyond recognition, and as a result, DNA analysis was required to correctly identify the victim's gender and identity. DNA testing can also be useful to determine pedigree information in regards to disease, or to better understand a breed of an animal. A case study that utilized DNA testing to investigate disease wanted to understand the spread of brachyspina syndrome, or BS, in Chinese Holstein cattle. BS is a hereditary disorder that causes early-term abortion or stillborn calves. Using the PCR-based genotyping method in Chinese cattle, BS could be traced back to a common ancestor, and as a result, recommendations were made to prevent further loss to the breeders. DNA testing can be used to understand also a dog's pedigree, to know how large the dog will grow, or to prove the dog that appear to be of a prohibited species is not one proven by DNA testing. There are many advantages to DNA fingerprinting that make it a great forensic tool. When DNA is tested, it is highly unlikely that 2 people would have the same DNA. Although human genes are identical between any 2 individuals, there are portions of DNA that do vary substantially. DNA sequences are varied in the number of repeats in individuals. This is due to the sequence of DNA base pairs. DNA fingerprinting is also important in the clinical setting. Bone marrow transplant DNA analysis has a large role with treating people that have cancer. DNA profiles are evaluated in order to determine if the potential bone marrow transplant would be accepted or rejected. The profiles are viewed by comparing the size of the DNA fragments by electrophoresis. Another advantage to DNA testing is that it can be used for tracing genealogical ancestry. Attached to this presentation are my own results from an ancestry test that I had done last year. Advantages of having this test done is to get an estimate of where my ancestors lived going back hundreds of years. DNA testing can also be used to assist in understanding how genes influence individual health. In this slide is my own DNA report I had done to assist in understanding how genes can influence my body size and weight, ability to lose weight, ability to maintain weight loss, the way that the body stores and processes dietary fats, the likelihood of altered blood triglycerides and cholesterol, and I was able to use this information to make better informed decisions about my own health management. As part of this DNA test, they also provided dietary recommendations based on my genetic type. This was done via studies that shown that individuals that share the same genetic results responded well with the suggested diet, which improved their health and weight. Thanks for listening. I will now hand over to Corina, who will now talk to the shortcomings of DNA fingerprinting and the potential future applications. Thank you.
Thank you, Megan. Hi, everyone. My name is Corina Necu, and I'm going to discuss about the shortcomings and potential future applications of the DNA fingerprinting. As with anything, there can be shortcomings with this technique, and the most common one would be the human error through incorrect sampling or application of the technique itself. For example, if you think of a crime scene, samples will most definitely be contaminated with factors from the environment, and therefore this process of DNA profiling will take longer. Moreover, it is known that DNA fingerprinting through gel electrophoresis requires larger sample sizes. When compared to the atomic force microscopy method, the results indicated that this technique could be a better alternative in terms of sampling and calibration. Consider the context of forensics. Sometimes larger sample sizes are hard to procure, and therefore other techniques may need to be considered. Even though DNA fingerprinting is conducted by machines and computers, at the end of the day, comparing two DNA profiles from a crime scene is still done by a human. And therefore, as Dan Crane interactively explains in his TED Talk, and I've included the link below, the downside of this method is that the final result can still be biased by the examiner. And Mr. Crane really explains this really well, and it's quite interactive presentation, so I would strongly encourage you have a look. As my colleague Samantha mentioned earlier, DNA fingerprinting's main principle is protein staining, which can be chromatic blue dye, silver staining, fluorescent dyes, radio labeling. As Dos Anjos et al. 2011 explains, protein spots can have different absorption thresholds and given the fact that human implications of the technique, errors can occur. So Dos Anjos et al. Really raises the concern of contamination with dust particles during the procedure. Now, what could be some potential future applications or extensions of this technique? The use of DNA databases have become a very controversial subject nowadays due to the ethical and privacy concerns. The advantages of using such a system is that they could reduce criminality and help police solve cases such as Craig Herman's case a lot faster. Mr. Herman was convicted of manslaughter in 2004 after his DNA was sampled. The blood found on the brick thrown at the victim's car was compared using a national database, and initially no matches were found, as Mr. Herman did not have a criminal conviction. But one of his relatives did, and that's what the UK Forensic Science Service did. They used a new technique of DNA fingerprinting to trace down Mr. Herman based on his relative's DNA. This genome matching technique has proven to be more cost-effective and as accurate as conventional DNA fingerprinting. Initially, it was used for identifying matching donors for bone marrow transplants. The GMT works by organizing genetic information into polymorphic frozen blocks, which are more stable and less susceptible to recombination. So there's a lot of research on that nowadays, and I encourage you to have a look online, see what you can find. The DNA fingerprinting can also be applied to a concept that's quite interesting called food forensics. The term of food adulteration represents the process of modifying a food's structure, such as adding water and sugar and other ingredients with the aim of obtaining a similar product with similar physical properties. Now, it sounds a bit complicated, but it's quite straightforward, and you can see it nowadays in a lot of products. It's when they add sugar, water, and flavoring agents to obtain more orange juice out of one single orange, for example. And there's been some serious health implications that have been identified because of food adulteration, such as development of food allergies. I'm sure you would know one of your friends that has developed the food allergies, and this could be because of this, or even more serious than that, drug interactions. So therefore, food forensics nowadays is looking into identifying these types of foods appropriately, and it's quite an important subject. Now, the table below shows the top 25 ingredients that were target to food adulteration processes present in scholarly records dataset. And it's quite concerning to see olive oil and milk being the top two, as both these ingredients are part of our daily diets. Now, probably one of the most controversial cases of food adulteration, which I'm sure many of you know, was the Subway chicken story, which became viral overnight when CBC released an article saying that Matt Harnden from the Trent University's Wildlife Forensic DNA Laboratory proved that the poultry samples taken from a Subway restaurant contained only 50% chicken. Now, even though, and I've looked in all sorts of databases, I could not find reliable evidence to sustain this. Even that, this shows us the importance that DNA fingerprinting could have in today's market. So I hope you really enjoyed our presentation, and thank you so much for listening.